Welcome to Principal Component Analysis in Python. Principal Component Analysis, or PCA, is a popular way to understand and simplify complex datasets. Let us discover how PCA helps us find patterns, reduce dimensions, and visualize data. We will use real examples including the famous iris flower dataset. Running the cell now. What is PCA? PCA finds patterns in data by looking for the directions where the data varies the most. It lets us make a big, complicated table of numbers into a small and simple one, so humans and computers can see important shapes. Running the cell now. Let us prepare Python by hiding extra warnings. This comment reminds us that we want less distractions from warning messages. We import a special warnings library. It helps control how warnings show up in our results. This command tells Python to filter out all regular warnings. That way, we can focus on learning. Running the cell now. Now we are ready for clean outputs. Try adding this to any notebook when you want less warnings. It is time to see some real data. This is a comment marking our data setup section. We load the iris dataset using scikit-learn's built-in loader. Loading means getting the data ready in memory. We import pandas, a library for handling tables called data frames. It is very common in data science. We keep the main data in a data frame called X, using the feature names from the dataset. The species label, what kind of iris each flower is saved as Y. Printing the shape tells us how many rows and columns the data has. Now we print the shape, which shows the size of our data. We print the first 5 rows to preview real values. This shows the first 5 examples as a table. Running the cell now. Try using .shape and .head on other data frames you make in the future. These give a quick checkup on your data. Seeing the class distribution is important when exploring data. This is a comment to show what comes next, checking groups in the data. We type out that we are going to print species counts. This sets up the output for the user. We use value_counts on Y. This counts the number of samples for each iris species. Running the cell now. Balanced classes help methods like PCA. Try value_counts with different label columns to check balance. Knowing the numbers range and spread helps us with PCA. This comment reminds us why we are running describe, to get quick summaries of numbers. Describe prints the count, mean, standard deviation, and other statistics for all features. Running the cell now. Check if some numbers are much bigger than others. That can affect analysis. PCA works best when features use a similar scale. This comment tells us we are about to standardize, or rescale, the data. We import standard scalar for this task. We create a scalar object. It knows how to shift and shrink our data. Fit_transform adjusts X so each feature is centered at 0 with standard deviation 1. Here we print mean values for all features after scaling. This confirms the centering step. Running the cell now. Standardizing helps prevent larger numbers from overpowering smaller ones. Try it on your own datasets. Starting PCA. Let us start finding the important directions in our data. We will use scikit-learn, a well-known Python library, to do the math for us. Running the cell now. Now let us use PCA to study directions of maximal variance. This line sets up a comment to explain what we are doing. We import the PCA tool from scikit-learn's decomposition module. Here we create a PCA object, ready to learn from standardized data. We fit the PCA model on our scaled features. Fit means the model will find the main patterns. We print how much variation each principal component explains. This helps us understand which directions matter most. Running the cell now. Try running fit on other datasets. Seeing explain variance guides you in choosing how many components to keep. Visualizing the results helps see how much information each component keeps. This comment introduces a plot for cumulative explain variance. Now we import matplotlib.pyplot for plotting charts. Charts make patterns easier to see. We plot cumulative sums of explain variance as we add each component. The marker draws a circle at each point. The X label tells us how many components are used. This label shows what % of original information each step keeps. We give the chart a helpful title. Finally, we show the plot to see how quickly we can represent most of the original data. Running the cell now. Look for points where the curve flattens out. That tells you enough components might be kept there. Let us compress our data down to 2 principal components for easier visualization. This note preps us to use only 2 components. That helps when visualizing. Here we create a new PCA object set to keep only 2 components. We fit and then transform the scaled data with 2 components only. We check the shape after the transformation. There should now be 2 columns. Let us see the first few rows of new PCA values. Printing the transformed rows helps confirm the size and contents. Running the cell now. Try other values in n_components to see how data size shrinks. Plotting the transformed data helps spot groups and patterns with your eyes. This explains we want to make a scatter plot of PCA data. We set a figure size for a clearer display. Here we loop 3 times, once for each iris species and a color. We draw dots for one species at a time, using colors for clarity and easy reading. We add labels to the axes for context. Labels improve understanding of what each direction means. A legend shows which color goes with which species name. A title gives viewers context for the chart. Finally, we show the full chart to visualize the species separation. Running the cell now. Practice with color and marker choices to make your plots easy for anyone to read. Try visualizing different splits or clusters. It is important to know what each principal component is made of. This is the part where we look under the hood at the directions PCA finds. Printing the main directions lets us see what features are most important in each new axis. The matrix here shows how the original features combine to create the 2 principal components. Running the cell now. Try to link these numbers back to the original features. Bigger values mean a bigger influence. Let us see how close we can get to our original data using only 2 components. This explains our new goal, to reverse the PCA process and return to the original space. By applying inverse_transform, the data moves from the compressed 2-dimensional form back to the original space. Let us look at the first few reconstructed rows. Printing the numbers helps us see how much is preserved and what is lost. Running the cell now. The more components you keep, the closer your reconstruction will match the original data. PCA can help models run faster and sometimes better, especially on big or messy data. Here we set up to combine PCA with a classic classification algorithm. We import tools for splitting data and for logistic regression, a common starter for classification. We split the reduced PCA data and the labels into training and test sets. The random_state gives repeatable results. Now we create a logistic regression model and fit it to training data. We fit the model, which means it learns to link features to labels. We check how often our model is right on the test data. Printing the test accuracy gives us real feedback on our pipeline. Running the cell now. Try changing test_size or algorithms. PCA can help both people and models spot the most important patterns. Sometimes, you may want to test with just one component. Let us see how much information that keeps. This comment introduces a mini experiment. Use only one principal component. We create and configure PCA for just one direction. We fit and transform the data, compressing everything to a single value per sample. Check the first few results after this dramatic reduction. Printing helps us compare with previous outputs. Running the cell now. Try plotting the one-component results in a histogram. You can check how much the data shrinks. PCA only works when all features are numbers. Let us try a fake column of text as an example problem. This comment gives us a key rule: PCA does not handle text. We label what we are testing next. We make a copy of X to keep our original data safe. Adding a fake text to column simulates a real data mistake. A try block lets us safely see what error comes up. We attempt scaling with the bad data on purpose. If there is a problem, we print the error message. Running the cell now. Always double-check that your tables for PCA only have numbers. Unexpected types can make your code crash. Mini project, reducing a real dataset. Now you try. Pick another dataset or use Iris and walk through these steps. 1. Load the data. 2. Standardize the features. 3. Run PCA for 2 to 3 components. 4. Visualize in a scatter plot. Tip: try the wine dataset with scikit-learn or a CSV from the list above. Running the cell now. Pause here and type out these steps on your own. Practice makes patterns stick. Recap. PCA helps us explore and simplify big sets of numbers. Always scale your features before running PCA. You can visualize main directions for data patterns. PCA is used in visualization, cleaner data, and even pre-processing for machine learning. Try out PCA with your own data to see patterns appear. Running the cell now. You are doing great. Try sharing your results online or with a friend. Thank you for learning PCA with us. Please like and subscribe to our channel for more beginner Python and data science tutorials. Let us keep growing our coding skills together. Running the cell now.